Hi there, welcome to this video. So we're going to be looking at proteins now. So proteins are made of amino acids, so they're polymer molecules. And the monomers that they're made of are amino acids. Now there are 20 different amino acids, and they're different because they have something called a different R group. AA's amino acids. So proteins are polymers, they're made of monomers. The monomer that they're made of is amino acids, and there's 20 different amino acids that they're made of. So every protein has a specific shape, and the reason it has that shape is because it's got a specific sequence of amino acids. Okay. Now, proteins, we tend to say their function is for growth and repair. Now that's a bit of a simplification because actually proteins are used for lots and lots of things. For example, enzymes are proteins, and enzymes are needed to catalyze every single reaction. Hormones are proteins, so that's our chemical messengers being sent around our body. We've got loads and loads and loads of different proteins. Some of them are structural, so things like collagen, muscle, etc., etc. So we've got loads and loads and loads of different proteins, but the thing they have in common is they're all polymers made of the monomers amino acids. Okay, now what we're going to do is we're going to start off with looking at the structure of an amino acid. So you need to be able to draw this off by heart. So it's got a central carbon, and carbons always have 4 bonds. At the top, we've got a hydrogen, and at the bottom, we've got the R group. Now, the R group is just representing something that you don't need to know. So, for example, the R group could be CH3. The R group could be just a hydrogen. The R group could be, however, something really huge. So it could be like a carbon ring with all these bits coming off it. It could be a huge structure. Okay, but you don't need to know what it is, but you need to be ready to recognize a diagram that has all the other features that you know, and possibly a letter R or possibly something drawn that you don't recognize. Now it's called an amino acid. And the reason it's called an amino acid is because on the left-hand side, we've got an amino group or an amine group, which is NH2. Okay, so NH2, you can see one N and 2 H's, and that's called an amine group. And that's why it's called an amino acid. That's where that word comes from. On the right-hand side, the group is an acid group. Now, it's called carboxylic acid, the same as we have on the end of a fatty acid when we're talking about lipids. And a carboxylic acid is COOH. And we draw that with a C, a double bond to the O, a single bond to the OH. Now, sometimes in an exam, they will simply ask you to circle where the amine group is, or to circle where the carboxylic acid group is, or simply to circle where the R group is. Remember, the R might not be written as an R, it might be written as whatever it actually is in that particular amino acid. There are 20 different amino acids. So when 2 amino acids join together, we call that a dipeptide. So we've looked before at how the prefix di means 2. So it's 2 amino acids. So you have to know how those 2 amino acids bind together. Now, the bond that they form between them is called a peptide bond. So you can see the words are kind of repeating themselves. So it's a dipeptide and it's a peptide bond. When we join lots together, you'll see in a minute, it's called a polypeptide. Okay, and you can also think P for protein, P for peptide bond, P for dipeptide. Okay, so all the P's go together. So let's have a look at how we form this dipeptide bond. So the first thing is we need to draw 2 amino acids side by side. So have a go at doing that. See if you can draw it from memory. So remember the hydrogen above, the R group below, the amine group on the left, NH2, and the carboxylic acid group on the right, C double O, double bond O, OH. or you can just say COOH, or to describe the drawing of it, C double bond O, single bond OH. Draw another one, carbon in the middle, hydrogen above, R group below, amine group on the left, and carboxylic acid group on the right. Now it forms during what we call a condensation reaction. Condensation reactions, like condensation on the wall, on the window, forms a molecule of water. So we circle these ones here. This is where the water molecule is going to form. We can write plus H2O down here. And then we can rub those out because they're involved in making the water. They have gone, and the peptide bond simply forms between that carbon and that nitrogen. The peptide bond is all of this. Right, so that's a peptide bond. And that's how we join two amino acids together. Now, the convention is that when we've done that, we then move that oxygen just up to there and that hydrogen up to there. Okay, it's just the convention how we draw them rather than leaving them on the diagonal. Remember to leave the double bond for the oxygen just there. Okay, so let's go through next the levels of structure in building a protein. So I always draw it in this four-way grid. So we've got 4 levels of structure: the primary structure of a protein, the secondary structure of a protein, the tertiary structure of a protein, and the quaternary structure of a protein. Sometimes you might see them simplified as primary, like that, the secondary, like that, the tertiary, like that, and the quaternary, like that. It's a simplified way of writing it. First, second, third, fourth. So the primary structure is the sequence of amino acids. And the amino acids, as we just learned, are joined by peptide bonds. And that chain there is called a polypeptide. So we can say in a polypeptide. And to draw that, we could simply, very simple, just draw it as a line like that. The secondary structure is the localized folding. Now, localized folding means just some parts of the polypeptide fold. So here's our straight polypeptide. Now, some bits of it might fold into an alpha helix, and other parts of it might fold into a beta pleated sheet, just a back and forth zigzag. Some parts, like this part here and this part down here, haven't folded into anything. We need to know a little bit about these structures. So this one here is called an alpha helix, and this one here is called a beta pleated sheet. So we need to know those 2 examples of secondary structure. Now, those structures, the alpha helix and the beta pleated sheet, are held together by hydrogen bonds. So here, holding that alpha helix, and here, holding that beta pleated sheet, are hydrogen bonds. And you need to know the types of bonds. So we've got peptide bonds in the primary sequence, and we've got primary structure, and we've got hydrogen bonds in the secondary structure. Okay, now those hydrogen bonds, a little bit about them. If we go back to our picture of the amino acids, let's say just here. Okay, I'm just going back to this just to save me time. If I had two amino acids, let me just finish off those pictures. So it would be NH2, and that'll be COOH. The hydrogen bonds form because some of these parts of these molecules have a slight charge. Now I'm going to use a little symbol that looks like this. This means slight. It's delta, Greek letter delta. It means slight. So what actually happens is the double bond O actually has a slight negative charge on it. Okay, and the N to the H there actually has a slight positive charge on it. And positive and negative attract, so between them, we're going to get hydrogen bonds. All right, so even if we had that peptide bond in there, Okay, you'd still have the O sticking up there, the H sticking up there, and it doesn't have to be two that are next to each other. It can be two in the polypeptide chain that are 20 amino acids apart. But wherever you've got this oxygen, it's slightly negative. Whenever you've got this hydrogen, it's slightly positive, and we get a hydrogen bond forming between them. Okay. The hydrogen bonds, let's just put a heading here, hydrogen bonds. The hydrogen bonds are called an intramolecular force. Now, intramolecular means within the same molecule. So they're actually forming within the same polypeptide. Not between different molecules, but within one molecule. So for example, if we had our alpha helix like this, and obviously all the way down these are amino acids that this is made of, it might be that you've got the double bond O sticking out there, which is slightly negative, and then you've got the nitrogen there, which is slightly positive, and therefore between them you'll get a hydrogen bond holding that helix in the shape. So it's within the same polypeptide chain, it's within the same molecule. Now, hydrogen bonds on their own are very weak, but because there are so many, they've got basically strength in numbers, strength as there's very, very many of them. So because there's lots and lots of them, it makes the structures nice and strong. The next level of protein structure is the tertiary structure. Now, in the tertiary structure, our polypeptide chain folds up into a big 3D shape. It will still have elements of alpha helix, elements that are folded into beta pleated sheet. Overall, it's now got an overall 3D shape. And you need to know the bonds, the different types of bonds that hold that shape. Oops, sorry, I was just trying to make a little bit more space. There we go, that's better. So the bonds that hold that 3D shape that we need to know, excuse me. First of all, we've got hydrogen bonds. So hydrogen bonds will form between any part of the molecule where you've got slight positive and slight negative, and you'll get a hydrogen bond. We usually draw hydrogen bonds as dotted lines as they are the weakest type of bond. Then you get ionic bonds. Ionic bonds form between parts that are totally positive and totally negative, not slightly positive and slightly negative, but totally positive and negative. And we draw those as a single line because they've got the middle strength. And then finally, we've got something called disulfide bridges. Now, the disulfide bridges form between two sulfur atoms. So if we've got a sulfur and another sulfur, we'll get a disulfide bridge. And we tend to draw those as double lines because they are the strongest of the three bonds. Now, these sulfur atoms, you only find them in one type of amino acid, and the amino acid that contains sulfur is called cysteine. Remember, there's actually 20 amino acids, and it's only cysteine that contains sulfur. So wherever you get a disulfide bridge, it'll always be between two cysteines, so it's between two sulfurs. The other thing that helps this tertiary structure to form this particular shape is that wherever we've got let me just make a bit more space. We don't need much for Quaternary. If we move all of that across, wherever we've got, if I use a highlighter, hydrophilic side groups, they attracted to water, and therefore they're going to move to the sides of the molecule. Okay, so hydrophilic are always going to be attracted to water and therefore will move to the sides of the molecule. Hydrophobic R groups, on the other hand, repel water and therefore they will always move into the middle of the molecule. Okay, so the hydrophobic side groups will always move into the middle. And this is a really, really specific 3D shape that it forms. It's very, very important. The protein will only be able to do its job, have its function, if it's that exact right shape. And all those bonds work together to form that shape. Now, the reason it forms that shape is because of the order of the amino acids in the primary structure. So the sequence of amino acids in the primary structure means that the R groups are in specific places, and therefore the bonds all form in specific places, and the protein folds up into this specific 3D shape. Now, quaternary structure just means that the protein is made of more than one polypeptide. Now, some proteins have a quaternary structure, and some don't. They don't have to. All right, but some of them will have more than one of these polypeptide chains, perhaps 3, perhaps 4, all joined together, and then they're said to have a quaternary structure. If not, they just have a tertiary structure. So all proteins have a tertiary structure, but only some have a quaternary structure. Now, once the proteins are kind of finished, then they start to form an overall shape. And there's basically 2 different shapes that they can form. The first one is called a fibrous protein, and the second one is called a globular protein. So fibrous proteins are long, extended shapes, something like that, possibly made of helices wound around each other, or they could be straight chains. Globular proteins fold into something that's roughly spherical. Okay, so we call this an elongated shape, and we call this a spherical shape. So the shape is very, very important. Okay. Now, a famous globular protein is hemoglobin. Now, hemoglobin is made of 4 polypeptide chains, so it has a quaternary structure, 2 alpha chains, which I'll draw in one colour, and 2 beta chains that I'll draw in another colour. That alpha and beta is not the same as the alpha helix beta pleated sheath. That's something different. This is just chain A and chain B. It's just the names they give them. So hemoglobin is an example of a globular protein. Now, this protein has we call prosthetic groups in it. So within these 4 polypeptides, it has some little non-protein groups. Now, non-protein groups are called prosthetic groups. Like if you had a prosthetic leg, it's like it is part of you, but it's not made of you. So these prosthetic groups are part of the protein, but they're not made of protein. So the prosthetic group that we get in hemoglobin is heme, and it contains an ion that carries oxygen. So one hemoglobin molecule has 4 polypeptides, therefore 4 heme groups, 4 ions, and therefore can carry 4 molecules of oxygen around. Okay. The fibrous proteins have this elongated shape, and this gives them the feature that they're insoluble. Globular proteins tend to be soluble. They dissolve in water. Fibrous proteins are insoluble. There's no way this huge long structure is going to hide in between water molecules and dissolve in water. So they're insoluble proteins. And the famous one for this is collagen. Now, collagen is a fibrous protein that's structural, and it actually has a really repetitive sequence of amino acids. This protein here, this hemoglobin, there's 20 different amino acids, and it has a whole selection of the 20 in a specific order. Whereas collagen has this repeating unit with loads of glycine and proline amino acids in. It's got a really repeating pattern of amino acids in it, and it forms these long helices. Okay, so we find it in our connective tissue and with our ligaments and tendons. And basically, it's just there for strength. It strengthens various parts of our body. It's a really long, thin structure made of a few helices wrapped around each other. And it's actually made of 3 chains. Not every fibrous protein is made of 3 chains, but collagen, the example that we need, is made of 3 chains wrapped around each other, making it really, really strong. Now, the length of these chains, obviously, they're chains of amino acids, and it's made of about 1,000 amino acids in a line. So it's really, really long. In between those 3 helices, it It has little hydrogen bonds. Remember how we talked about the hydrogen bonds holding the alpha helix? It's a similar thing. These hydrogen bonds are very, very weak on their own, but together there's so many of them that they actually make the molecule really strong for its structural form. The last type of protein that we need to know actually combines the last two topics. It's called a glycoprotein. Sorry, glycoprotein, and we have looked at them in the cell membranes. So the prefix glyco refers to something to do with carbohydrates, to do with sugars, okay? And obviously protein means protein. So a glycoprotein would be some kind of protein molecule like that with some kind of carbohydrate like glycogen like sort of protein, carbohydrate, sorry, sticking out the top. So that's your glycoprotein, and the glyco is a carbohydrate chain. Remember, carbohydrates have glycosidic bonds. That's another way to remember it. So a glycoprotein is a molecule that's made of carbohydrate and protein joined together. Okay. The point at which they join, remember this protein is actually a polypeptide, all folded up into a specific shape, which is a chain of amino acids. This point where the carbohydrate joins onto the protein is it will be joined onto the R group of one of the proteins, okay, of one of the amino acids in the protein, sorry, to fix that carbohydrate onto there. Now, they have lots of functions like structure, reproduction, immune response, hormone, protection of cells. But one of the important things is about the property of them. So these carbohydrate chains are actually hydrophilic, which means that they always move towards water. So if we had a cell, for example, we might have a protein in the cell membrane and it might have a little carbohydrate chain sticking out. So we've got a glycoprotein there and it'll always move to the outside of the cell and stick out because it's hydrophilic. Okay, it can go out into aqueous environments. Okay, and a lot of the time they're used for things like cell signaling, etc., because they're like this little recognition thing sticking out the side of the cell, telling other things what that cell is there for. All right, and that brings us to the end of our proteins topic.